Hello and welcome to New Scientist Weekly, your one-stop shop for the most fascinating science and technology news. I'm Christy Taylor in New York.
And I'm Timothy Revel in London. This week on the podcast, we'll be making sense of the latest decisions around 3 promising drugs for Alzheimer's disease, and if they are really as promising as they first seemed. Plus, we'll be talking about the mystery of the banana-shaped galaxy, if problematic smartphone use is a real condition, and using the moon to store Earth's most valuable biological samples.
But first, what was the first life on Earth actually like? The suspicion has long been that it was single-celled, something resembling bacteria, but much more primitive. But, barring the very unlikely invention of time travel, our only hope of finding direct evidence is to look in the oldest rocks on the planet, for fossils, just like with other ancient life. And sure enough, biologists on just such a quest have found what appear to be fossilized cells of primordial organisms. But these cells are really strange and nothing like what they expected to find. If, again, we're presuming that the earliest life looked something like modern bacteria. But now one team has managed to recreate a lot of these strange fossils and shown what kind of life might have made them billions of years ago. Michael LePage reported on this for us. Hey, Michael.
Hey, hello.
Can you start by telling us, first of all, about these ancient fossils? Why are they so mysterious and controversial and strange?
So we're talking here about the time not long after Earth formed. That's around 3.5 billion years ago. So there are not many rocks left from that time that might have preserved fossils inside them, but there are now quite a few studies claiming to have found fossilized cells in these ancient rocks. The thing is, these supposed fossil cells are really odd. So lots of them are much bigger than modern bacteria, like 70 times bigger. And some of them also appear to have internal structures which bacteria don't have.
Okay, so biologists were expecting to find these tiny and simple fossil cells left by, again, something like ancient bacteria, and instead they found big, complex ones left by something different?
Exactly. So there's a lot of controversy about what these fossils are. Some people think they aren't the remains of living cells at all. After all, 3.5 billion years is a very, very long time for traces of cells to remain preserved. But even those people who think they really are fossils of early life haven't really been able to explain why they are the way they are.
Until now, right? I mean, you're kind of leaving us in suspense and keeping this tense, but there's now a potential explanation, right? So come on, what is it?
Yes, indeed. So it turns out that these fossils maybe can be explained by modern bacteria in a way. Now, to explain that, I need to say just a little about what bacteria are. I'm simplifying, but essentially they're bags full of liquid. They don't have the same kinds of internal structures that the more complex cells in our bodies do. But what bacteria do have is this rigid outer cell wall that maintains their shape. So if you imagine getting a bag full of water and sticking it in a box, that's pretty much what a modern bacterium is like in terms of structure. Now, the thing is, what happens if you kick a bacterium out of its box? Now, there are some extreme conditions where bacteria can lose their cell wall, and then you just got this sort of shapeless bag of liquid, and usually it swells up and it bursts, and that's the end of the bacterium. But if it's really salty, in these conditions, these naked bacteria, as it were, can sometimes survive and even reproduce. And these things are called L-forms because they were first discovered here in London at the Lister Institute. Now, the thing is, people have known about these L-forms for decades. What's new is that a team has put a load of these L-forms in conditions just like those that might have existed on early Earth. And what's happened is that these L-forms have produced structures that look really remarkably similar to a lot of these mysterious early fossils. So they grow as big and they look very much the same shape and so on.
Okay, this is pretty amazing. So it kind of looks like an L-form, grows like an L-form. Could it actually be an L-form? Is that what the earliest life was like? Maybe kind of bacteria without a cell wall?
Exactly, that's the idea. So maybe these fossils represent primitive ancestors of bacteria that hadn't yet evolved a cell wall, but these L-forms are behaving in the same way as those early primitive ancestors. So it's a really appealing idea because it explains a lot of puzzling findings. L-forms divide in all kinds of odd ways that change depending on the environmental conditions, for instance, how salty it is. And in some conditions, you have dozens of new cells forming within a single L-form, and then this big parent cell eventually bursts apart and releases them. Now, it's a really messy and inefficient way of dividing because that parent cell loses some of its liquid, but it also creates what look like complex cells with internal structures, explaining some of these bizarre fossils. Now, by comparison, Modern bacteria with their cell walls divide very efficiently. They divide neatly into two. But of course, you wouldn't have expected the first cells to just come into being with the ability to control their shape and to divide neatly. It's much more likely that the first cells weren't very good at these things and that they were rather haphazard and out of control.
Okay, so we're explaining these strange fossils as maybe bacteria that aren't very skilled at what bacteria are now very skilled at. Is that kind of the idea?
That's exactly it.
Amazing. So this is a case of one mystery around the origin of life solved, maybe. We now know absolutely 100% for sure what the earliest life looked like. I know this is a trap.
Yes, of course. So all this team have done is stick some L forms in different conditions and show they can reproduce the shapes that we see in these, what we think are these early microfossils. So that's very far from proving what those things actually are. But I think these findings are really striking, and I think a lot of people are going to want to follow it up and do a lot more studies. So I think this is the start of a whole new avenue of research.
Now, we're meant to be in a new era for treating Alzheimer's disease. In the last 3 years, US regulators have approved 3 new drugs to treat the symptoms, the latest of which, called donanemab, was approved in early July. Along with the approvals came serious excitement about this being a turning point for Alzheimer's. These drugs, they work differently to those that have come before, and remarkably, 2 of them are the first shown to actually slow the condition's progression, rather than just easing its symptoms. And this, of course, should be good news. However, over the last 3 years, there has also been calls for caution, with some saying the drugs are not as impressive as they first seemed. And just last week, an advisory body to the European Union has actually recommended against approving one of the drugs. Alexandra Thompson has been keeping a close eye on all this for us and is here to help explain the situation. Hi, Alex.
Hello.
Okay, so there's a lot to talk about here, but could we just begin by you giving us a bit of a reminder about these 3 drugs? What are they and how do they work?
Yes, so as you mentioned, the US has approved 3 drugs for slowing the progression of Alzheimer's disease. These are donanemab, lecanemab, and aducanumab. So other than the US, only the United Arab Emirates has approved aducanumab, while Japan, South Korea, China, Hong Kong and Israel have authorised lecanemab. And donanemab has only been approved in the US. So all 3 of these medications use antibodies that have been engineered to target and remove amyloid proteins from the brain. So the clumps or plaques these proteins form are believed to be critical in driving the progression of Alzheimer's disease, which is known as the amyloid hypothesis.
Right, and under this hypothesis, if the amyloid plaques are causing Alzheimer's symptoms, then removing them should relieve or at least slow some of the symptoms from getting worse.
So that's what the hope was, but in clinical trials, only lecanemab and donanemab were found to actually slow the rate of cognitive decline in people with early stage Alzheimer's. So after 18 months, people on lecanemab had a 27% slower rate of cognitive decline compared to the control group on a placebo. But for those on donanemab, cognition declined to almost a 30% slower rate. So this may or may not sound significant to you, but when these percentages are put into context on a scale that measures quality of life and symptoms among people with Alzheimer's, the difference of treatment versus placebo is only just a few points, if that. So the question and the controversy is whether that difference actually translates into an improvement that someone with early Alzheimer's themselves would recognise, or if their caregivers or their loved ones would pick up on it. So, but as you say, the first drugs to show an effect on the condition's progression after decades of research, that's why some people are calling them game changers.
Yeah, and I mean, given we've so long had basically nothing for Alzheimer's, 30% does sound like it makes a difference in someone's life. But what about that first drug you mentioned, aducanumab?
So the clinical trial data on that is conflicting, which is why its approval in 2021 was so controversial. The drug's manufacturer Biogen actually halted clinical trials in 2019 after early evidence showed the drug had no effect on cognition. But 7 months later, it said an analysis of new data from a subset of participants who were specifically receiving a high dose showed an effect. So it was eventually approved, but an advisory panel recommended that it wasn't going to be approved. So there's murky evidence and there are also side effects, which is what's giving people pause.
When we're talking about the side effects for a drug like this, how serious are we talking? What's going on?
So all of these medications carry a risk of brain bleeding and swelling, which is known as amyloid-related imaging abnormalities, or ARIA. So most of these instances are asymptomatic, you don't even know you have it, and they resolve on their own. But in some cases, people can experience a headache, a seizure, or even death. So with aducanumab, about 40% of the participants experienced ARIA, and so far 4 related deaths to that drug have been reported. And 11 people also died during phase 3 trials, but Biogen has said that none of those could be attributed to the drug.
Okay, so certainly a little controversial, perhaps not as much evidence of benefit and risk of some serious side effects with aducanumab. But what about the other two? There's lecanemab and donanemab, and there was certainly a lot of excitement around those. Are those equally as risky?
Unfortunately, yes, they come with risk too. So for lecanemab, clinical trial data found that about 3% of participants who were given the drug developed severe brain bleeds, and sadly 3 of them died, though the drug's maker, IZ, says it didn't cause the deaths. And with donanemab, about 6% of those taking the drug in trials experienced this severe brain bleeding, which sadly contributed to 3 participants' deaths. So some experts say that the benefit of these medications is pretty small, so is it worth the risk of these serious side effects? This is especially true for aducanumab, given there's so little evidence it can affect Alzheimer's progression or improve quality of life and cognitive decline and all the rest of it.
I mean, this all feels like really quite the come down from that initial excitement around all these treatments. Is there some positives here?
Well, there's positives in the sense that Alzheimer's disease is very challenging, and unfortunately there hasn't been a lot of progress in terms of medications despite decades of research. But to add to the bad news, these cost tens of thousands of dollars per person per year. They require lengthy infusions. They have to be administered in the early stages of the condition because they're not effective if Alzheimer's is more advanced. So the fact that 2 of the drugs have been shown to slow its progression, I mean, that does give some hope. It's almost like the very beginning of the end, because finally we have a treatment that really does slow that decline, even if there are all these caveats.
Let's take a quick break to tell you about another podcast that I think you'll really like. Karan Rajan is an NHS surgeon, a medical professional even, and in his podcast, Dr. Karan Explores, he pursues answers from the most knowledgeable guests out there on some of the most pressing questions in health, from why medicine has been so neglectful of women's health to the ABCs of having a healthy gut microbiome. Sleep, caffeine, even psychedelics, it's all there.
His interviews are delightful, insightful, and humorous. Whether you want to understand the newest frontiers of research or just get some insights into things you could do differently, Dr. Karan Explores is there for you.
You can listen by searching Dr. Karan Explores in your favorite podcast app.
And also, we'll soon be interviewing psychologist Lucy Fawkes about her newest book, Coming of Age. It's all about adolescence, what science can tell us about the lives and behaviours of teenagers. We'll get behind the stereotypes, bust some myths, and dig into what the last decade of research is revealing about this critical age.
And we want to help answer your questions. Do you have teenage children? Are you a teenager? What do you wish you understood better about teen behavior, emotions, and even social situations? Or are you trying to make sense of your own teenage years, however long ago that was? Send us an email to podcasts@newscientist.com, or even better, record us a short voice memo on your phone and email us that. That's podcasts@newscientist.com. Think about a galaxy. Any galaxy, ours, someone else's. Odds are good you've got a big, relatively flat, spirally mass of stars in your head, like the Milky Way. But that's not the only shape a galaxy can take. Some are more round, for example. But either way, it was noteworthy to astronomers when the JWST started taking pictures of the universe a couple years ago and found some galaxies that were very distinctly shaped like, well, bananas. This was exciting to scientists, actually, and Alex Wilkins is here to explain what these banana-shaped galaxies may tell us about the early universe. Hey, Alex.
Hey.
Okay, so I've said the word banana a few times. That's a weird shape for a galaxy. What's going on?
Yeah, so to understand this puzzle, we first need to understand a bit about how galaxies form, or how astronomers think galaxies form. There's this standard model for galaxy formation, which assumes that most galaxies start out as fairly symmetrical disks before evolving into these more complex swirls like our own Milky Way. So when we use telescopes to look at light from the early universe, we would expect to see basically big round clumps of stars.
But
In the 1990s, the Hubble Space Telescope, which was the predecessor to the James Webb Space Telescope, started spotting these elongated, or what astronomers called prolate galaxies in the early universe that didn't quite fit with this picture that they'd imagined. There was lots of debate about whether we were actually seeing what these galaxies were formed and were actually like. So these shapes could have been caused by some sort of gravitationally warped light, for instance, with matter in between us and the galaxies, or it could be a consequence of Hubble only being able to see certain kinds of galaxies because of its sensitivity. Most astronomers thought these shapes were kind of interesting and they were intrigued by them, but we couldn't definitively say what was going on because of Hubble's shortcomings.
And I feel like now, like we've said many times before, JWST comes to the rescue.
It strikes again, exactly. So last year, astronomers looked at some pretty empty parts of the sky, and again they started seeing loads of these elongated galaxies, except this time JWST was much more sensitive, so they couldn't use that same argument that Hubble's weak sensitivity was like an observational bias. There was still the possibility of gravitational lensing, but now those same astronomers who found these prolate galaxies with James Webb last year have done an analysis to show that the level of warping isn't enough to produce the elongation. So these prolate galaxies really do appear to be shaped, funnily, like bananas. And if confirmed by other research, prolate galaxies are going to require astronomers to completely rewrite their galaxy formation models.
Oh, wow. Okay, so a complete rewriting the very process by which something like a galaxy forms. This sounds huge. Do we have any idea why prolate galaxies, these banana-shaped galaxies, might actually be shaped that way?
So yeah, it gets even more interesting. The same researchers who found these prolate galaxies also analysed whether the galaxies were pointing in random directions or whether they lined up in any sort of pattern. And they found this statistical signal that they were being aligned by something. Now, there are two obvious ways this could happen. It could be because there's some dark matter that we don't know about in the form of galaxies between us and these prolate galaxies that makes them look aligned when we observe them from Earth. But we don't know of any large galaxy clusters that might be responsible for that kind of lensing in those regions of the sky. The other explanation would be that these galaxies are actually forming along filaments of dark matter in the early universe and revealing the really complex structure of the early universe that is otherwise invisible. You can think of it a little bit like light bulbs that are kind of illuminating this hidden web.
Wow, that's such a wonderful picture. Given there's so little we know about dark matter, I'm also assuming the structure of this web would be real interesting and helpful to understand better.
Exactly. Astronomers and cosmologists would really like to understand this. So the distribution of dark matter in the early universe is super important for all sorts of cosmological reasons. Scientists can use it to feed back into our cosmological models, which make predictions about how fast our universe's expansion is accelerating or how quickly galaxies form and grow, for example. So if you know this structure, you could take a simulation, run it forward in time, and try and find the right combination of values that produce these galaxies and webs of dark matter. It's really a way to confirm our theories about what dark matter might be.
It sounds like there's some really impressive and big ramifications of this, if true. But we've still got that if in there potentially. There's part of this that still needs to be verified, that the weird-shaped galaxies really are as weirdly shaped as they seem and not just some trick of the light. How close are we to being certain about all of this?
You're right, this isn't set in stone yet. One researcher I spoke with who wasn't involved with the work said it is really exciting that these banana galaxies might actually be banana-shaped, as they appear to be, but that we've only seen a small number of them with JWST so far. We will need to observe many more with both JWST and future galaxy surveys before we can be sure that there aren't still observational biases like we were talking about earlier. So there's still a lot of work to be done here.
Since the dawn of the smartphone, people have been wondering if smartphone use is bad for us. I, for one, feel like I'm glued to my device way too often and I spend too much time using it. I'm addicted to my smartphone in a very colloquial sense. And if I was perfect, well, maybe I'd be a bit better at putting it down. But that, of course, doesn't mean I have a medical condition or an actual addiction to my smartphone. But that also doesn't mean such a condition doesn't exist. Now, that condition, called problematic smartphone use, is controversial. It's not currently recognised as an official condition by many official health bodies, but 2 new studies use the term in their attempts to understand smartphone use and mental health issues.
So what do we know about problematic smartphone use, and is it a real condition? Biomedical reporter Carissa Wong has just joined the team and is now joining the podcast. Welcome back to the New Scientist family, Carissa.
Hello, everyone.
Before we talk about these 2 new studies, how are researchers currently defining problematic smartphone use? Like, what is it?
So, according to researchers, someone with problematic smartphone use might feel as if they've lost a sense of control over their phone use. They might also be preoccupied with being on their smartphone, and that might be at the expense of meaningful activities in their lives. And they might also feel distressed when they can't use their phone. As Tim mentioned before, though, it is worth saying this isn't yet widely accepted as an official medical condition. It's not recognised by the World Health Organization, the US Centers for Disease Control and Prevention, or the National Health Services in the UK.
Okay, so talk us through the new studies. What are they about? What are they telling us?
Sure. So these two studies both link what they term problematic smartphone use among teenagers to increased symptoms of anxiety, depression, and insomnia. In one study, the researchers surveyed more than 650 teenagers between the ages of 16 and 18 about their smartphone use. Then they used something called the Smartphone Addiction Scale to score their usage. Participants with scores above a certain threshold were classed as having problematic smartphone use. The researchers found that 19% of these participants qualified as having problematic smartphone use. And those individuals were twice as likely to report symptoms of moderate anxiety, and they were also 3 times as likely to report symptoms of moderate depression. And that's compared with their peers who didn't have so-called problematic smartphone use. The team did account for factors that may influence the results, such as how long the participants said they spent on their smartphones each day. On that point, interestingly, they found that using your phone more wasn't linked to anxiety or depression symptoms, but this problematic smartphone usage was.
Okay, so that's interesting. I've definitely always been under the impression that I just need to use my phone less, not really change the way how I use it. So that is interesting. And what about the second study?
Yeah, so in the second study, researchers surveyed smartphone use among 69 teenagers. These were younger, so they were aged from 13 to 16 years old. The team also asked the teenagers about their experience of anxiety, depression, and insomnia symptoms. And they found in this study that 44% of the participants had problematic smartphone usage. And that's based on the same addiction scale as they used in the previous study I mentioned. So when they surveyed the participants a month later, they found that an increase in severity of problematic smartphone use over that time period was actually linked to more severe symptoms of anxiety, depression, and insomnia.
So what should we take from this? There's this general sense that I think many people have that maybe smartphones should take some of the cop for rising anxiety levels, especially among young people. But is that actually justified with this new research?
Well, it's important to note that these studies were small and they don't prove a causal link between smartphones and mental health declines. Also, just a minority of the participants were classed as having problematic smartphone use. Researchers I spoke to say that if there is a causal link between smartphones and mental health, it could either be that smartphones increase symptoms of anxiety, depression, and insomnia, or it could be that people with symptoms of anxiety and depression or insomnia are more likely to use smartphones in a more problematic way. Of course, it could also be both. We don't have enough evidence yet to say which.
Even as we start to untangle this, I'm sure there's still many people who would just really want to reduce their smartphone screen time, whether their use is problematic or not. What does the research say on reducing how much we're on our phones?
Yeah, there's certainly some research out there behind that. And in surveys of the participants who took part in the first study I mentioned, 95% of the participants had tried to limit their smartphone use in some way. According to those teenagers, the most effective strategies were occasionally putting their smartphone on Do Not Disturb mode, turning off notifications, or leaving their smartphone in another room at bedtime. For teenagers in particular, the researchers I spoke to recommended that rather than imposing harsh restrictions on teenagers' phone use, parents and schools could have more two-way discussions with them about what aspects of smartphone use are benefiting or harming them.
We now know categorically that there is no cheese on the moon. Nothing alive or even slightly biological that hasn't been left behind by an astronaut. But if one research team has their way, we could one day use the solitude and deep cold of the moon's polar regions to keep an array of DNA safe, including the microorganisms critical to cheese production. Reporter James Woodford has been taking a look this week at a proposal to construct a lunar biorepository where cryogenically frozen living cells could be stored. Hey there, James.
Hey, Christy, how you doing?
I'm pretty all right, and I'm very curious. So I have one very big and important question for you. Since most of us, including probably me, won't ever likely walk on the moon, does preserving your cells in a biorepository on the moon sound like maybe a good consolation prize?
Christy, I'd still much prefer to go to the moon myself, but I do actually think the scientists are onto an intriguing idea. I have to admit, when I saw the research paper proposing this, which was led by Mary Hagedorn at the Smithsonian Institution in Washington, D.C., I thought it sounded a bit kind of gimmicky and nuts, but when you look into it, there is some sense to it.
Yeah, I mean, when I think of biorepositories that might be really, really safe for a long time, I'm definitely thinking about the big seed bank at Svalbard, which is, purposefully in a cold and remote place. Why would we put a biorepository, even of very important samples, on the moon instead of places like Svalbard?
There's a few reasons, but the biggest is that in some of the polar regions, deep inside craters, it's permanently dark and always around minus 200 degrees Celsius. And this is important because to cryogenically freeze living cells requires temperatures that remain constantly below minus 196 degrees Celsius, which is the point at which nitrogen becomes a liquid. This means that there'd be no need for power to store the frozen specimens if they were going into storage on the moon. It's essentially just set and forget. There's nowhere on Earth even close to that kind of cold. And there's some other important considerations also. The first is that a biorepository on the moon is apocalypse proof for any catastrophe on Earth, especially from, say, climate change, geopolitical upheaval, or even just a simple disruption to the power grid.
Yeah, we've definitely had those power disruptions enough already as it is. I mean, even looking back at the Svalbard seed vault, I remember that it's had issues with flooding, power outages, other problems with the infrastructure. So yeah, let's look at the moon. How would it work? It seems like it's a pretty difficult place to get your samples there, but is there anything else that might make this hard to implement?
Yes, there's one really big downside, and that's radiation. The Earth's magnetic field and atmosphere protect us from most of the harmful radiation in space, but that is not the case on the moon. This means the biorepository will need to be shielded not only once it is established, but also all the material will need to be carefully protected from radiation on its journey from Earth. The scientists say the easiest way to shield the samples from cosmic rays is for them to be at least 2 meters underground.
All right, so let's say we get all this together. We figure out the radiation issue, and we're ready to store some cells in perpetuity on the moon. What kinds of cells are we talking about? What are we focusing on preserving? Are we talking like elephants? Are we talking about the blue whale? What's going on?
So this is where the microorganisms that make cheese come in. The idea of storing these living cells is not just have a zoo of charismatic animals like elephants, but also all of the microbes and agricultural animals, fish and other species that may one day be useful. Hagedorn says the project has 3 main goals. The first is to safeguard the diversity of life on Earth. Second, to protect species that might be useful for space exploration, such as those that could provide biomaterials for food or filtration, and to preserve microorganisms that may one day be needed for terraforming other planets. The researchers acknowledge that there is much that they still don't know and huge technicalities to overcome, even like just how big a biorepository on the moon would be. Each sample may fit into, say, a 1 milliliter bag, the scientists suggest, and Hagedorn says the total size could be as small as the size of a volleyball.
Wow, that is a very different version than, say, like the biblical Noah's Ark. So do other scientists think this is a good idea, or is it all Hagedorn on her own making this call?
There's agreement that such a repository is possible if enough willpower and resources are thrown at it. But some researchers think it will be extremely impractical. For example, there are a lot of questions about how researchers could even access the site, withdraw samples for use or study, or even add new ones. Other researchers, such as Alice Gorman at Flinders University in Adelaide, Australia, say that it would be worthwhile, even just as a kind of archaeological site for future civilizations, or maybe even extraterrestrial life, should they exist and find us, as a snapshot of life on Earth and our technology.
That's it for this week. If you like this show and think more people should know about us, please do give us a 5-star rating or even a review on Spotify and Apple Podcasts.
We've got more fascinating stories from New Scientist. You can find them on our website at newscientist.com, or just break open your closest 3 billion-year-old rock and see what's inside. We'll be back next week. Bye for now.
Bye.
This podcast is produced by OG Podcasts. Find out more at ogpodcasts.co.uk.